Hey, everybody, we are wrapping up Unit 3 today. And like I have mentioned before, we are going to talk about how changes occur in genetic info, what happens when DNA copying goes wrong or doesn't work. So, before we talk about that, I want to talk about how we control when DNA is used for transcription and translation, when that protein synthesis occurs, right? And the biggest way that our bodies do that, how organisms do that, are through operons, right? Now, previously we talked about how genes have these introns and exons, the parts that make it into the protein and don't, which parts make it into the mRNA and which parts don't, rather. And operons are the ones that code for the protein. They say, yes, this code will be part of that protein. The way that we kind of turn those off and on are called transcription factors. There are a whole bunch of transcription factors. We're going to keep it simple. Right? I want you to know that there are things in those exons that are usually removed that impact if the operon is transcribed or not. They kind of turn it off and on. Right? And I also want to talk about these special kind of genes called Hox genes. Hox genes code for body layout, and turning them off and on does some really weird things with growing parts of an organism in different spaces, right? So you can see the Hox genes for a Drosophila, which is a housefly, and for a mouse and a human. We have similar ones because we have similar body layouts. Our heads are at the front or top, 4 legs or arms, sort of a tail. You have a tailbone, they have a full tail, right? I just want you to see how these are kind of lining up. And later in the year, in Unit 6, we're going to talk about evolution. Evolution is going to argue that these genes are going to be similar, and they may even be very, very similar or the same at a DNA level. And they're going to say, oh, well, our bodies are even coded the same way. They're almost designed. They're not going to use that word design, right? But really, we're just seeing that God uses the same patterns across multiple species. These are going to be very different when we go farther apart in things versus different kinds of animals or even into plants or insects. But I wanted to kind of call them out. They are color coded in this diagram, so you can see how different parts of the Hox genes or different Hox genes on the same chromosome can impact these. The human one, it's on chromosome 6, 11, 15, and chromosome 2 that will together code for which parts of a fetus which parts of an embryo will become which parts of a fetus, which become different parts of babies, right? So, as a review, I want you to go back in your notes and check and remind yourself which part of a gene, which we're now going to call an operon, all right? So, which part of that will be, is it from the introns or exons during transcription? So, make sure you wrote that down, right? But when we are talking about DNA changing, we're talking about a mutation, right? The mutation is just any change in the DNA sequence that affects genetic information. We don't care if it's on an intron or an exon, any change whatsoever that affects that genetic information, even if it doesn't cause a difference in the actual protein that's coded for. So, mutagens are what cause those things to happen abnormally. It might happen as a mistake during DNA copying, but those are usually caught and filtered out in that in process of error checking. But exposure to certain chemicals, especially heavy metals, all right, or chemotherapy that you might have heard of, radiation causes mutations. You've probably seen sunburns. That's the most common way that we experience radiation mutation. As the damaged DNA needs to be removed, those cells die off, and you get like peeling, right? We can also experience mutations from infectious agents. All right. So bacterial or viral infections can cause mutations to DNA, and so they're labeled as mutagens. Depending on if that mutation is in a somatic cell, a body cell, or a germline cell, a gamete that's going to go on to make offspring, they're going to be different impacts, right? And I want you to think about those different impacts and tell me which one you think will have a bigger impact if it became mutated and why you think that, right? Write that in a complete sentence. That's our question, our second questions. I know it's labeled question 1, it's question 2. You can fix it in your notes, right? As we talk about different types of mutations, I'm just going to have you write down these tables. They are very self-explanatory, but we will be talking about them more in class. So click pause, write all of this down, then write all of this down, right? Please note that these are frame shift mutations, so they're going to shift the reading frame, which 3 nucleotides show up in a codon, right? And then these are going to be across multiple chromosomes. They are much rarer, but they do happen for inversion and translocation, right? So make sure you have all of those down in your notes. I will grade your notes back in class as we continue talking about mutations. I'll see you there.